Hey everybody, sorry it took a little bit longer to get this up than I was thinking it was going to. Here we go for the last chapter here. This is chapter 11. We're looking at just the chemical and physical control methods for controlling microbes in the environment. So again, just to remind everybody, we are really looking at, in this case, trying to control these microbes, keep them really out of the environmental areas where we don't want them. So this is really going to look at trying to get rid of, like it says there, cells, endospores, protozoa worms, the stuff like that, but again, out in the environment. And to me, this chart is probably one of the most useful ones here. It's kind of showing you that we're going to look at physical versus chemical versus mechanical agents here, and really looking to see how we can use these to remove microbes from the environment. And really, it's going to look at a few basic definitions that are going to be important here. And again, it goes back to what we talked about even with the antibiotics as well. There is these things that have much higher resistant, like the endospores and prions, those that are a little bit harder to kill, and then most growing cells that are pretty much the easiest ones to kill. So to kind of get you some of the basic definitions out of the way, and these are ones to me that are very important that you guys understand the difference between. So first one is going to be sterilization, and this is one that is going to get rid of any viable cell whatsoever. So this would kill your normal growing bacteria, but it's also going to kill endospores, everything else like that. This is an absence of microbes in that environment. Disinfection is a little bit different than that. Disinfection, you're removing most of the growing pathogens, but it is not necessarily going to get rid of everything. So disinfection is not sterilization. It is a reduction in the number of pathogens or reduction in the number of microbes, but not a complete absence of microbes. An antiseptic is really just what it is. It's a disinfectant that is used on the skin surfaces. Sanitation is mechanically, so like scrubbing them off, and then degermination is it's an overarching kind of term that's just saying reducing the number of microbes on something. So microbial death, we're really going to be looking at factors that can influence this a little bit. So things that could really alter what is going to happen with these microbes is if the population was larger, these disinfectants might take a little longer to work. What type of microbes are they? Do they have capsules? Do they not have capsules? What are the environmental conditions? Is the temperature ideal for these microbes, or is it not ideal? Is their pH ideal or not? These are all things that can influence how well a particular thing is going to work. What concentration you use this at? So if you don't use the correct concentration, you might have a problem getting this to work as well as you'd like. And then again, biofilms, other things like that being present. And other things to think about when we're really talking about this is if you're using a particular disinfectant or you're just sterilization, some type of technique here, when you're looking at any particular item and what you should do with it, so whatever we're going to be kind of disinfecting or whatever else, does it actually need to be sterilized? If it does, then we have to really look at these sterilization techniques. It's an item that's reusable. A lot of ways to get around this stuff is to use sterile, one-use type of things. And then also, can it withstand heat? Is it heat sensitive, moisture sensitive, radiation sensitive? All these things are ways to sterilize, and if it can't withstand those things, these are other considerations we're going to have to look at. Again, suitable for whatever we're working at here, ability to penetrate in, all that good stuff like that. And how these antimicrobials work outside the body, they're not way different than what we talked about with the antibiotics and how they worked inside the body. So what do they target? They target the cell wall. They target cell membranes. They target DNA and RNA synthesis stuff. They target protein synthesis. So really when it comes down to it, not necessarily protein synthesis, but interfere with ribosomes, things like that. So they're really working almost exactly the same way we were talking about with using the antibiotics and what they were doing. It's just showing you some examples that we've already actually kind of seen. So denaturization is one of these main things that a lot of these are going to do. And that's one of the things I would say to pay attention to when we're talking about these different methods. How do they work? What are they suitable for? Are they sterilization techniques? Are they disinfection techniques? Those are the things to kind of make sure you understand on each method that we talk about. So we're going to look at the physical control methods first. So the first one we're going to talk about is heat. And heat, there is actually moist heat and dry heat. Really the big difference here, and I used this example when I was talking to my other class as well, It's the difference between moist heat and dry heat. It's kind of one of those ideas that if I put you outside in 32-degree weather, you'd probably be able to last quite a few hours, things like that. It wouldn't be fun. It'd be cold, but you could last quite a while. Where if you fall into 32-degree water, that a lot of times is going to lead to hypothermia and death within 10 minutes. And it really has to do with the fact that water is a good conductor of heat. And because of this, these moist heat methods are a lot of times going to be able to use much lower temperatures and shorter exposures in order to really have their effects. Where dry heat, you're generally going to use higher temperatures and longer periods of time. And this shows an example on the slide right here with the one weird exception, this stereothermophilus, which is a weird one. But everything else you can see here, 121 takes 1 minute. With dry heat, it takes 2 hours. So you can kind of get the idea that certain ones here, it is going to be a little bit longer with this dry heat, longer and usually higher temp. And a couple of things to look at when you're talking about the thermal death here. A lot of times then there is formulas that do all of this that we're not going to mess around with. But the thermal death time, what this is, it's really the shortest length of time required. If I give you a particular temperature, this is kind of saying how long is it going to take to kill all these test microbes if given, let's say, 80 degrees Celsius. How long is it going to take? Where the thermal death point, it's not looking for an amount of time. It's actually looking for a particular temperature. So this is saying you have 10 minutes. What's the lowest temperature that will kill everything? And these are a couple of really easy type of definition questions that are easy to ask about. So first moist heat method, and this is actually a sterilization method, is autoclaving, probably the most widely used sterilization technique that's out there. On this one, it is steam under pressure, so we can get the temperature above 100 degrees Celsius, because just boiling water, that's as hot as it will ever go. But if you can do pressurized steam, you can actually get it above 100 degrees Celsius. And this is one that at about 15 pounds of pressure, temperature is going to be 121 degrees Celsius, and usually about 15 to 30 minutes is enough time to really sterilize anything that we're talking about. Main thing with this one is if it's moisture sensitive, that's going to be a problem. If it's heat sensitive, that's also going to be a problem. So again, it has to be things that can actually withstand the moisture and the heat that we want to sterilize to use this method. The other thing here is how it works, and it really is denaturing proteins. It's basically cooking whatever the microbe is. And this shows you an example of kind of right here, the tabletop one. And you can also get these big ones like this. A lot of hospitals now have automatic ones where you go up, hit a particular button, and it goes through the whole process. Tinilization, I wouldn't spend a lot of time, if I was you guys, worrying about this one. This is a way where if you couldn't autoclave it, it's a way to supposedly try to sterilize it. I wouldn't really call it a sterilization technique. I would call it a disinfection technique. What you do is you heat stuff up, kill off the vegetative cells. If there's any endospores, you re-incubate it, let it go back and go to vegetative cells. You repeat this 3 times. And what that is supposed to do is get rid of all the ones that were in spores and bring them out. But to me, there's no guarantee. So this one to me is not a sterilization technique. It is actually a disinfection technique. Boiling water, a lot of you have probably heard the idea of boil stuff in water for 10, 15 minutes and that will pretty much clean the stuff off. It is a pretty darn good disinfecting process. The only thing I would say about this one is anything that's an endospore forming bacteria, boiling in water is not going to kill it. Outside of that, it will kill just about anything else. Pasteurization, we talked about when we did the history here. Pasteur came up with this process as a way to kind of get rid of food spoiling organisms without destroying the food. He started out with this process right here where you heat up a whole vat of something up to the 63 to 66 degrees Celsius, hold it there for 30 minutes, and that's usually good enough. Nowadays, most of what they use for at least anything that's not like a mom and pop kind of cider mill or something else like that, they're going to use this flash method where what they do is they run it through a piping and they heat it up to a hotter temperature, but only for 15 seconds. And what they have is they have a tube that wraps it around a heating element, then it goes over to a cooling element, brings it right back down to temp, and again, it's going to get rid of most of the food spoiling organisms. It will not get rid of endospores really at all, so this wouldn't work for that. They actually have a sterilization technique that they go up above 100 degrees for about 2 seconds, and that's called ultra high temp sterilization, and it's where you get, if you've ever seen the shelf milk, or a couple of my kids have to drink rice milk, and but it's one until you open it, you don't have to refrigerate it. Like I said, dry heat is usually higher temperatures than moist heat, and usually longer times. Incineration is really good for like cleaning off inoculation loops or things like that, or instruments where you can heat them up red hot. The main thing with incineration is not going to be anything that's heat sensitive or a food product, because basically what you end up with is dust of it. Dry ovens, these are more like the oven at your house, heating up the temp, but this is one is that 150 to 180, that is the temp it has to get up to, in some cases up to 200 Celsius, and it's usually for about 2 hours at temp to get sterilized. And that 2 hours is not just set the timer 2 hours, put it in, and 2 hours later take it out. This is 2 hours at that 150 to 200 degrees Celsius at temp. So it doesn't count the heat up time, and it doesn't count the cooling down time. So it is a lot longer process to sterilize that way. And that's just kind of showing you an incineration technique there with these electric heaters for cleaning inoculation loops. Cold is another method. This is a good way to inhibit growth of microbes. You guys all use this at home. This is your fridge and your freezer. Main thing about this one, it is going to stop the growth a lot of times of microbes. What it will not do is actually kill them. So if you have something that's contaminated, you put it into the fridge, you bring it back out and let it sit out, it's still contaminated. Same thing with the freezer. You can go all the way down to the temperature of liquid nitrogen, which is 200 degrees or more below freezing, and if it goes in contaminated, it will come out contaminated. Another thing that kind of works, and this is kind of like the beef jerky example I always use, desiccation. If you dry stuff out, a lot of times there's not enough moisture in there to allow the growth of these things to happen. It's part of the reason if you have like a bag of Jack Links, you could probably leave it out for about a year and it's still probably going to be fine. This is why they used to salt meats when they would take stuff on the wagon trains across West. Just a way to kind of preserve the stuff that's in there and really limit stuff from growing by the fact that you are pretty much taking all the water out of it. Radiation is another physical method. You're pretty much using, in this case, there's a couple different types of radiation. Ionizing radiation is a lot of times when you guys think of radioactive things, that's the idea with ionizing radiation. So X-rays, gamma rays, things like that. What happens, these can penetrate into the organism. Honestly, they can penetrate into us. They mess with the electrons. They break apart DNA, and it is honestly a very good sterilization technique for especially food products and medical supplies, widely used nowadays. The other type of radiation is the non-ionizing radiation. And this is showing you the example of the electrons breaking up the DNA. The non-ionizing radiation, this is UV radiation. So some of what's in the sunlight, it goes in there. It does actually mess with the DNA as well. Main thing on this one is it has to come into contact with the light directly. The microbe does. Even if you had a sheet of paper between the UV source and the microbe, it's not going to penetrate through, and therefore it's not going to harm the microbe. Filtration, to me, is another physical method here. You're really just, everybody knows what a filter is, like a coffee filter. It stops the coffee grinds from going through, but it doesn't stop the water from going through, and it's just based on pore size. I think the next slide kind of shows you the idea here, where you have this stuff not being able to go through the holes, but the liquid can. There is actually sterile filtering, so certain liquids that you can't heat up, or air units, like it says here, you get small enough pores and air and water and liquids can pass through, but not any microbes, viruses, or anything. So you can actually sterilize using filtration. All right, I'm going to escape out of this real quick here, guys, and do a second show in just a second.